Hello, my name is Taneshia Harmon, and I'm in your cell biology class. And the protein that I was giving was RNA polymerase. So let's go ahead and get started. RNA polymerase was first discovered by Sam West and Gennard Hurwitz in 1960. RNA polymerase is an enzyme, which means an enzyme, they're made up of amino acids, and the purpose is to allow the cell to carry out chemical reactions very quickly. So in a sense, it acts as a catalyst. But specifically with my enzyme, it is going to be responsible for transcription. What is transcription? Transcription is the process of creating a form of RNA from DNA with my enzyme, RNA polymerase. So DNA is it's not allowed to leave the nucleus. So the information has to be copied, and that's where the process of transcription begins. And transcription does take place in the nucleus. And there are three divisions of transcription. The first being the initiation division, which is requiring construction of the RNA polymerase complex on the gene's promoter. The second one is the elongation, which is the synthesis of the RNA transcript. And the third is growth, the finishing of RNA transcription and disassembly of the RNA polymerase complex. So what I want to speak to you with next is what actually happens during transcription. And first, the RNA, which is one-stranded, remember RNA is one-stranded and DNA is two-stranded. So the RNA bonds to the DNA, and since they are both hydrophobic, that allows that bond to take place. So the RNA binds with the DNA strand at the promoter, which is the specific sequence of the gene. And this unwinds and unlinks the two DNA strands using one of those strands as a template. So the RNA then matches the two nucleotides with its complements of the DNA strand. So guanine, if I said that correctly, guanine now is with cytosine, and adenine is now with uracil. So then they bind these two nucleotides together to form the DNA strand, which is now the messenger RNA, the mRNA. Then it stops when it encounters a termination sequence of bases. Then from there, the messenger RNA will go into the ER, where they will bind. Not bind. The ribosomes or whichever they'll build a new protein in the the ER. So that's pretty much what my enzyme is doing during this transcription process. As far as the structure of RNA polymerase, with it being relatively new, there actually isn't a lot of information on it. There haven't been a lot of studies done with it, but primarily the the protein structure is based off the primary, secondary, tertiary, and the quaternary levels. I honestly couldn't really find the breakdown of my protein as far as the levels, but I did find what the RNA is pretty much comprised of. And if you want to go back to that first slide I had with the three-dimensional rotation of my enzyme, the RNA polymerase, you can kind of follow along with what I'm saying. But one of the pieces of that is the clamp, and this clamp keeps the polymerase connected to the RNA, and it swings open and close, which changes the size of the active site channel, allowing more things to move in and out. So there is that level of selective permeability. Have to be the right size to come in, and that sort. There's also the flap, which ensures that the messenger RNA is held down and retained. Like, we don't want that moving during the transcription process, because if the transcription gets done improperly, then that'll cause, a further mutation within the process, and then the cell will either die, or we'll have to start the process all over again. And the last thing that I did find on it was the rudder, which prevents the DNA RNA hybrid from occurring. So you don't want the DNA and the RNA to transcribe improperly. So I did find that as far as the structure of my protein goes, but as far as the basis of RNA polymerase in its primary, secondary, and tertiary and quaternary levels, I did not find that information. Now I'm going to move on to the types of RNA polymerase. There are bacterial RNA polymerase, eukaryotic or yeast yeast RNA polymerase, and there's the archaeal RNA polymerase. But for my presentation and the paper that I wrote, it's on the eukaryotic aspect of it, because that's what we're studying in cell biology. So the RNA polymerase is broken down into 3 subunits, and the first subunit being polymerase 1. And this polymerase is pretty much responsible for the transcription of ribosomal RNA, which consists of 96% of RNA. So this polymerase is very active. There's never a down period, because we're always going to be doing something. And this polymerase actually is located in the nucleolus. Is constricted to synthesize at the same pace with growth requirements, development, and division. So they want it to all occur, on schedule, on time. They don't want it to be, fluctuating. It needs to stay at a steady pace. The next one is polymerase 2, which is the second subunit, and it transcribes messenger RNA and small non-coding RNA. This is found in the nucleoplasm. This is very, very important to the scientists, and this is where a lot of the studies are getting done, which is why there are vast amount of research and information on this polymerase. And the last subunit would be the third polymerase 3, which is responsible for transcribing the tRNA. I couldn't find exactly where this polymerase would be located, but I did find that it reads 3 messenger RNA bases, which are the codons, and places the corresponding amino acids at the site with the adjacent amino acids. So that's what it's responsible for doing. As far as any rare diseases or things of that sort with my protein, I didn't find any. I didn't even see anything about any diseases. The only thing that I found is that a mutation could occur within the transcription process if something were to happen where the RNA polymerase wasn't binded, or it didn't speed up the reaction as it was supposed to. But in general, my protein is very important for the transcription process, because a lot of things have to be duplicated, and we can't even go into the third stage, which is the translation stage, without the transcription phase. So as far as genetics go, RNA polymerase plays a very big role in that process. So that is RNA polymerase. Thank you for taking the time to watch my video. If you have any questions, please shoot me an email at tjh019@gmail.com or shsu.edu. Thank you, and have a great day.